So now, we all know the structures we have to shove glucose into our cells. Now we talk about what we actually do with it. The first step is glycolysis, the initial splitting of glucose. Glycolysis occurs in the cytoplasm of our cell. For future reference, all of these reactions are enzyme-induced reactions, but we would need an entire 6-season TV show of characters to name them all, so we're going to skip that. An ATP molecule finds itself into the glucose and uses the energy it releases to attach its phosphate to the glucose, a 6-carbon molecule. This happens again, and we get a super unstable 6-carbon molecule that exists for an incredibly short period of time. The molecule splits itself into two G3Ps. The G3Ps get a hydrogen and 2 electrons ripped off of it and are attached to an NAD+ molecule, creating NADH. The G3P also gains, then immediately loses a phosphate to make ATP, and becomes PGA, 3-phosphoglycerate. It's stripped of another phosphate, making another ATP, and becomes a major product of glycolysis, pyruvic acid, or pyruvate. To recap, glucose needs 2 ATPs to turn itself into 2 G3Ps. Each G3P makes an NADH and 2 ATPs. So our products are 2 NADH and 4 ATP, net 2 ATP. The next step is often looped in with the Krebs cycle, but it's its own step entirely. This is our first aerobic reaction. The previous step could happen in the presence or absence of oxygen. The pyruvate from glycolysis is taken into the mitochondria and to the matrix of the mitochondria. It is here that CoA, coenzyme A, is attached to the pyruvate. This rips off a carbon which bonds with some oxygen and floats away to be exhaled. This also releases a hydrogen and 2 electrons, which is promptly scooped up by an NADH+. The major product of this is our acetyl-CoA. The Krebs cycle, the almighty NADH factory. The acetyl-CoA from before sheds that enzyme and combines with oxaloacetate to make citrate, which is why it's also called the citric acid cycle. We happily rip off a carbon to make more CO2 and an NADH. This means that we now have a 5-carbon molecule called ketoglutarate. But don't get too attached to him because a bit more of CoA comes along and we lose another carbon and make another NADH. So now we have succinyl-CoA. The CoA is shed and a phosphate from the molecule is attached to GDP to make GTP, which turns ADP to ATP. Plain old succinate now loses 2 hydrogen ions to turn FAD to FADH to make fumarate, who gains an H2O molecule to make malate and sheds its final hydrogen and 2 electrons to make one more NADH. But this time, no carbon is lost. This makes our molecule oxaloacetate, completing the cycle. The electron transport chain, the reason that we have all of this NADH in the first place. The electron transport chain is the name of the electron part specifically, but the whole thing is called oxidative phosphorylation. But when somebody says the electron transport chain, they usually mean the whole thing. This occurs in the inner membrane of the mitochondria. The membrane contains 4 complexes, 2 coenzymes, and an ATP synthase. As opposed to photosynthesis, the complexes are actually numbered in order. NADH turns into NAD+ at complex 1. It releases 2 electrons into the complex, and the hydrogen ions are released into the matrix. The complex uses the energy from the released electron to pump 4 hydrogen ions into the outer space of the mitochondria. The electrons are then pushed into coenzyme Q. Complex 2 is specifically for FADH2. It doesn't pump any protons, but it still gives electrons and releases hydrogen. Coenzyme Q takes electrons from complex 1 and 2 and donates them to complex 3. Complex 3 pumps 4 protons into the outer space. The electrons from 3 are taken by carrier C, who gives them to complex 4, and complex 4 pumps only 2 protons and turns the other 2 into water. All of the hydrogen ions in the outer space make for a large concentration gradient. The hydrogens move down the concentration gradient and spins the ATP synthase to bind the P to the ADP. It's also really important to understand that the final electron receiver of this entire chain is actually oxygen, which is where the water is made. So to summarize the products of each one of these steps, In glycolysis, we spend 2 ATP to make 4 ATP and 2 NADH. In the Krebs prep, we make 2 NADH. In the Krebs cycle, we make 2 ATP, 6 NADH, and 2 FADH2. And in the electron transport chain, we make 34 ATP. Now, what's also important to understand is that each NADH can make a maximum of 3 ATP, and each of the FADH can make a maximum of 2 ATP. These numbers aren't always exact because some energy is always lost in the process. So as you can see, this works out to 10 NADH making 30, and the 2 FADH coming in for the extra 4. So altogether, we make 38 ATPs. And that's the entirety of aerobic cellular respiration, minus the enzymes because who has the time for that?